Hi everyone, welcome to this week's Science Podcast and happy Autism Awareness Month. Yesterday, ASF celebrated World Autism Awareness Day by announcing 9 new pre- and post-doctoral fellowship awards to support autism research. I'd first like to introduce the ASF 2015 class of pre- and post-doctoral fellow awards. We received over 75 of these, making them incredibly competitive. The 9 represent an amazing mix of junior-level scientists who show promise for making long-term contributions towards the field of autism, together with a mentor that will help them complete a research project making an impact in the lives of people with autism. For the sake of presenting them in some sort of organized way, I'll group them into general categories. First, risk factors in autism, both genetic and environmental. William Brandler at UCSD is working with Jonathan Sabat to sequence the entire genome of people with autism. In addition to the sequencing itself, William is going to help develop methodologies to help analyze this data, which will be important for both this project and others. In further evidence that the genetics of autism is more complicated than scientists originally anticipated, Jason Keel will be working at the University of Michigan with mentor Kenneth Kwan to study something called somatic mosaicism. This means in rare cases, a mutation will show up in one type of cell and not others. This represents a totally new type of genetic finding and also one that could impair the ability to do genetic testing in all individuals and clearly needs more research. Finally, Natasha Morris will collaborate with John Constantino to study the recurrence of autism in female siblings of boys with autism. We get a lot of questions from parents about what the risk of unaffected siblings is to have kids of their own with autism. We really don't know this. This study will address that question. The only way to do this is through a large database. So the researchers will be going through the Interactive Autism Network and providing evaluations on children born to family members to determine if there's an elevated risk. The study will also help researchers understand what's called the female protective effect in autism. Now, no research findings or no discoveries come through just one study. Most require multiple studies which build on each other, and they all start in basic neurobiology. This is where new ways to treat and understand autism are discovered. They're sometimes unrecognized and unappreciated in autism research, but the Autism Science Foundation is strongly committed to this line. First, Marissa Coe, sorry, not Melissa, which is how I spelled it on the press release, at the University of Texas Southwestern Medical Center will be working on a mouse model to understand how some of the genes associated with autism contribute to how the brain forms. Another predoctoral fellow, Nathan Kopp, will be investigating the mechanisms by which oxytocin is released in the brain. We obviously know oxytocin has been considered as a potential therapy for autism, so this will help refine and examine this hypothesis even more. Finally, Daniel Nedelko at Mass General is focusing on the calcium channel. Now, this is particularly interesting because both genetic and environmental factors have been shown to influence the functioning of this channel. And the genes that we know that affect the function of this channel are also known to play a role in cognitive processes. So this study lays the groundwork for studies which target a mechanism as an intervention. And in the end, families want prevention or treatment strategies. ASF is supporting 3 fellowships in this area. First, in an unprecedented opportunity, Shou Wang at Caltech will be working with leading neuroscientist Ralph Adolph to capture brain activity directly from neurons in people with autism. There's a patient population with intractable epilepsy and autism that will undergo surgery for their epilepsy, and they'll be studied. There's no other way to gain this sort of access to brain cells in awake, functioning people. Jean-Paul Noel, under the guidance of Mark Wallace at Vanderbilt, is going to be doing some pretty extensive analyses on sensory integration in autism and how it ties to language and cognition, which will be the basis for future treatments for sensory issues in autism. Finally, we know that most people with autism don't receive just one therapy. They receive multiple therapies. Rachel Zamzow at University of Missouri, along with her mentor David Beversdorf, is going to study the combined effects of behavioral intervention in children with autism together with an anti-anxiety and drug propranolol. So congratulations to this amazing class of research fellows, and you can bet that we'll be reporting on exactly what they found when they find it. On today's podcast, I also want to highlight a news story about the long-term consequences of maternal immune activation. There's been some pretty solid evidence that infection or inflammation during pregnancy has a number of adverse outcomes in offspring. For example, it's been shown to produce behaviors in animal models that are similar to autism, like repetitive grooming. There's also been reports of increased risk of autism and schizophrenia after maternal immune activation. For example, epidemiological studies have shown an increased risk of autism in mothers who had a severe infection, mostly during the first or second trimester of pregnancy. So researchers have been studying this as a risk factor and have started to examine it in the presence of specific genetic risk factors. But what goes on in the brain of these individuals and how long do the effects last? A recent study from UC Davis examined this in a monkey model. They exposed pregnant monkeys to what is known as poly-IC. Poly-IC is a compound known to induce an inflammatory response in animal models. It's not a virus, but it's a compound that mimics the effect of a virus or a challenge to the immune system, and it's the same compound that's been used in many animal studies. The researchers studied the brains of offspring about 3 years after birth to see if the neuropathology was similar to that of what's been reported in human postmortem brain tissue studies. Multiple studies have reported that the brains of people with autism, there are more spines or branches from the trunks or bases of neurons compared to those without autism. In the brains of monkeys who were exposed to this immune challenge, the dendrites were thinner, and in some regions of the dendrites, they did see more spines. The effects were mild and actually more reminiscent of schizophrenia. But it was also noted in the discussion that the monkeys were juveniles, and in human tissue, most of the donors are from adults, so differences may have changed over time. We certainly know this to be true from human postmortem studies. The findings are still important for autism research because it shows that maternal immune activation has long-term consequences in offspring. Also interesting were behavioral results. Again, animals exposed to this challenge exhibited more stereotyped back and forth pacing in their cages, which is a form of repetitive behavior. Because this is a potentially preventative risk factor, the results will identify novel preventative and therapeutic strategies that may alleviate the structural changes in the brain seen with this exposure. And this is the perfect example of why knowing what the human brain looks like is essential to better understanding the risk factors of autism. Thank you all for listening, and if you haven't yet, please register for the ASF Day of Learning on April 22nd. Tickets are going fast, and we want to make sure there's a seat for you. Talk to you next Friday.